Hi, this is my little review video I'm doing since we're not having a chance to have our review day. I have a list here of topics that I use to make up the final exam. So I'm going to go over the topics in general just to help give you an idea of the things that you want to be sure you study. It's a 55 question test, online, just like the other ones. Not much calculations, a few images, but not many. Most of them are going to be multiple choice questions covering the topics that I consider the most important when I gave the lectures in the first place. So I would urge you, besides, looking at these or listening to this video and get a grip on the main topics, go through the practice questions, make sure that you can answer and particularly explain the practice questions. The PowerPoints, you can go through the PowerPoints and look particularly at the red areas and make sure you understand those, not just that you kind of remember it. Think back to the test that you took and topics that might have tripped you up on the last test and study those topics a little bit harder. It should be pretty straightforward. There's not going to be new material on this except for the reproductive section. There'll be maybe 10 or a dozen questions on the reproductive system that you won't have been tested on. The questions you'll see on the final rarely are the same question you had on your block exam. Sometimes they'll look the same, but, maybe on the block exam I said, well, which of the following would make blood pressure go up? And this time I'll say, which of the following would make blood pressure go down? And it'll be the same looking question, but you just have to remember to look for the right answer that would answer its new version. So there may be a couple of questions that are actually the same, but most of them will be very similar, but not the same. So let's start off with our little blood quiz we did, the blood section we did at the beginning. You want to know the types of blood cells there are. What do erythrocytes do? What do neutrophils do? What do eosinophils do? Basophils? What do you have the most of? Is their most likely job? Know the way that T lymphocytes work compared to B lymphocytes. We've seen a lot of that with the coronavirus. The T cells aren't going to really do us any good right now. What we need are the B cells to make antibodies so that our neutrophils then will properly attack the viruses and kill them. Or the antibodies could even bind to the virus and block it from attaching to the cell, to your own cells, so that it can't inject its DNA or something. But know the difference between B cells, T cells, memory cells. Memory cells are B cells that are prepared to make antibodies at the drop of a hat if that virus ever shows up again. The blood clotting system. Know the intrinsic and extrinsic cascades. Don't know the parts of it, but know that the intrinsic, is a cascade that is set off from internal damage, from external damage to a blood vessel. But they actually have to come together at some point to form the final clotting cascade that brings you thrombin and fibrinogen to fibrin, which is the actual clot. Understand that platelets don't form a blood clot. They form a platelet, little gooey blob. They platelet adhesion or not coagulation, but aggregation stops the bleeding right away, and then they send out signals for the real clotting cascade to make an actual blood clot. And aspirin blocks platelet aggregation. Heparin blocks the actual clotting cascade, and then TPA breaks it up after a clot is formed. So you can use that to break up an existing clot. Know that erythropoiesis happens especially in flat bones and irregular bones, mostly flat bones in the marrow, the bone marrow, that actually red blood cells, white blood cells, and platelets are primarily made in the flat bone marrow. The stimulus for erythropoiesis, or the red blood cell formation, is erythropoietin, which is made by the kidneys when they sense low oxygen levels. So if your oxygen goes down, your kidneys release erythropoietin, you make more red blood cells, and you can carry more oxygen. And that's what happens if you go to altitude and do altitude training. Know what hematocrits are. It's, a percentage of your blood that is red blood cells. Know approximate normal hematocrits, males a little higher than females. Know how to do blood typing. There'll probably be one blood typing question on there. If you have this, what can you take? If I already got it, I can take it. If it's anything I don't have, I can't. So practice a couple of those. Know the blood type, what could happen if you get a wrong blood type. If you get a Rh positive blood and you're Rh negative, that means you're getting an Rh factor you don't already have, and therefore you're going to have this coagulation or clumping of red blood cells that can kill you. And that's how erythroblastalis fetalis works. If a Rh negative mom has a baby with Rh positive blood, and especially after the first childbirth where a little bit of blood mixes, any subsequent child that's in the same condition, the mother will have made antibodies and try to kill that baby, thinking it's some kind of an alien creature. All right. Then we did the cardiac section. Start off knowing the parts of the heart. Know the blood flow in through the vena cava, right atrium, tricuspid valve, right ventricle, pulmonary valve, pulmonary arteries, pulmonary veins, all that stuff. Know that travel pattern so that if there's a problem getting from here to here, you know where the problem is. If you have a tricuspid valve stenosis where it won't open, what's going to happen? Blood won't be able to get from the right atrium into the right ventricle because that's where it is. Know the difference between the right side and the left side of the heart. Right side is pulmonary. It's pumping low pressure blood into the lungs. Left side is systemic and is pumping high pressure blood to the whole body. Know how to calculate cardiac output using heart rate and stroke volume. And remember that actual oxygen consumption is heart rate times stroke volume times extraction, which is AVO2 difference. So if you got this times this times this, it would just be this times this times this, would give you actual oxygen consumption. What determines your heart rate? Well, it's your SA node fires at its own rate without being stimulated by anybody. It can be accelerated and decelerated by sympathetic or parasympathetic effects. The parasympathetic primarily is the vagus nerve, which would slow it down. So your resting heart rate is actually lower than the intrinsic firing rate of the SA node because the vagus nerve, the parasympathetic vagus nerve, keeps it suppressed at rest. The vagus nerve leaves it alone during exercise, and sympathetics speed it up. Sympathetics not only speed up the heart, but make your heart contract more forcefully to increase stroke volume. Remember the Bainbridge reflex? If you get stretch in the atria, causes your heart to beat faster. It's heart rate. Starling reflex, if you stretch the ventricle, it makes the ventricle contract more forcefully to increase stroke volume. So increased venous return causes increase in heart rate and stroke volume to give you better cardiac output. Remember how it generally works when you start exercising. Your heart speeds up, but the sympathetic nervous system actually constricts all of your blood vessels worldwide. And then local factors cause dilation in the places you need it. So it increases the resistance maybe in your arm if you're only pedaling on a bike, and then locally you'll dilate those blood vessels in your legs so that blood is pushed into your legs where you need it. Know what creates the first and second heart sounds. Know a little bit about your EKG. You know, the isoelectric line is when nothing is happening. P wave is the atrial contraction. QRS is ventricular contraction. The ST segment, if it's up, you're having a heart attack. If it's decreased, you're having ischemia. And then the T wave is ventricular repolarization to get ready for the next beat. Know the factors that change from vessel to vessel. You have the smallest total cross-sectional area at the aorta, therefore the blood goes the fastest. At the capillaries, you have the largest total cross-sectional area, so the blood goes the slowest. And then it speeds up again. Pressure goes from the aorta, it's highest at the aorta, and it's lowest at the vena cava. Know approximately what those pressures are. Know how you filter blood out into the tissue and then reabsorb it at the other end with your Starling forces. Okay, hydrostatic pressures are high. You push out more blood. Hydrostatic pressures are low over here compared to oncotic pressures, so you reabsorb. The same situation we had at the kidney with the glomerular capillaries versus peritubular capillaries. Respiration. Know how ventilation works. The muscles of inspiration and expiration. Diaphragm is the main resting inspiratory muscle. It works without almost any other effects. Expiration at rest is passive. You just recoil back to your original size to blow out. And your externals can help you pull out, and your internals can help you pull in if you're exercising and need more power. Know how you change the pressure in your lungs compared to the outside to move air in and out. When pressure is negative inside compared to the outside, you're going to suck air in. When it's positive inside compared to the outside, you're going to blow air out. Know your lung volumes. Be able to add tidal volume, inspired, expired reserve, and get vital capacity. And then add your residual volume to get total lung capacity. Know how those various volumes work. Know how to calculate alveolar ventilation by taking out the dead space each time. Because it's simply your tidal volume, as many times as you breathe, minus your dead space each time. Because that's the air that doesn't make it into the alveoli. Know how the lung recoil, which is elasticity, counters compliance. Compliance is how easy it is to open your lungs. Elasticity is how hard it's pulling back. If they're perfectly balanced, you can breathe out and in as you should. With emphysema, you break all the rubber bands. You can fill your air with lungs, but you can't get it out, so they're full of stale air. Know how pulmonary surfactant works, coming from the type 2 cells in the alveoli, and therefore makes it easier to breathe in than it would be if you didn't have pulmonary surfactant. Know about the AA gradient. Remember the good old AA gradient. If your alveolar oxygen is too different from your arterial oxygen, you got a problem. You know, if they're both high, you're hyperventilating. Both low, you're hypoventilating. If they're too split, then you're ventilating okay compared to how much is actually making it into the bloodstream. Endocrine system. Know basically where everything comes from. Releasing hormones from the hypothalamus. Know which hormones, the tropic or stimulating hormones that come from the anterior pituitary, and the two neurohormones that come out the back. antidiuretic hormone and oxytocin. Know what stimulates their release, what they mainly do, each one mainly does, and what the negative feedback is. Okay, so you've got the pituitary ones, but then you also have the other ones we did, which were parathyroid hormone and calcitonin for maintaining calcium levels. And you have, we know we have our thyroid hormones and what they do and what hyperthyroid and hypothyroidism look like. Cortisol, too much is Cushing's disease. You know what that looks like. You know what too much growth hormone looks like. You get too big or you get too thick if it's acromegaly. Insulin and glucagon, how they work. Insulin puts sugar into the tissues. Glucagon takes it back out again, primarily from the liver, when you need more glucose in your bloodstream. Okay. Know the difference between aldosterone and ADH. This comes in handy with the kidney too. Remember, aldosterone is part of the renin angiotensin-aldosterone system. So it starts with the kidney, detecting low blood pressure, and it renin, angiotensinogen, angiotensin 1, angiotensin 2, aldosterone retains sodium and therefore water by osmosis, and your blood volume goes up. But it goes up isoosmotically because you're getting both sodium and water. And so that's the renin-angiotensin system. Whereas ADH is released from the hypothalamus, and mainly, if your blood pressure is low, you retaining water will get it up. But mainly, if your blood becomes too thick, if you sweat too much and your blood becomes too thick, that hyperosmolarity will cause you to release ADH so you retain more water. Antidiuretic hormone causes you to retain more water, your blood volume goes up, and it gets thinner. It decreases the osmolarity of the blood. Know how water-soluble versus fat-soluble hormones are carried in the blood by lipid-soluble ones are transported by transporters, as opposed to just being dissolved in the plasma, and they can generally go straight through the membrane, straight into the nucleus, and have their effects there. Know a little bit about diabetes mellitus, which is the glucose version of it. Diabetes insipidus is when you can't make enough ADH, so you pee too much and you're dehydrated all the time. Diabetes mellitus means that you have so much glucose in your blood that it overwhelms the kidney's transporters, exceeds the transport max, and you start peeing out some glucose. When the glucose stays in your pee, it osmotically draws more water, so you pee too much. You're thirsty all the time, you're hungry all the time because you can't get the glucose into your muscles where it's supposed to be, into your brain where it's supposed to be. It floats around in your bloodstream. Your blood sugar's too high. I mean, people who have diabetes, their blood sugar's too high. That's because it can't go in the tissues like it should. You either aren't making insulin, type 1, or you can't respond to it properly, type 2. Digestive. Know what happens at each place. What happens? What does saliva do? What are some of the things that it does? Review those. Know what salivary amylase does. Okay, it's the first digestive enzyme, starts out with starches a little bit, get digested some in the mouth. Mastication, chewing, deglutition is swallowing. It goes down, it goes through the upper and lower esophageal sphincters into the stomach. You got the fundus, you got the body, you got the antrum, you got the pyloric sphincter. The stomach is going to release, it's going to be stimulated primarily by protein to release gastrin, which will cause you to release hydrochloric acid, which will start the digestive process. It'll also stimulate and turn on pepsinogen into pepsin so you can start digesting the protein. So protein digestion will begin there a little bit. Primarily, though, as you move into the duodenum, you will notice the fats and proteins. You'll release CCK to give you more digestive enzymes, secretin to get more bicarbonate to neutralize your stomach acid. And then the final processes will be at the brush border with sucrase and lactase, which you can be deficient in, and so forth. Various proteases at the brush border. And then those various transporters that allow us to take in the glucose and I don't even know if I mentioned this. If I did, forgive me. But that glucose transporter, the glucose-sodium cotransporter, the identification of that was credited with saving millions of children who were dying from dysentery and cholera, every year, because they would, especially with cholera, what you do is you stimulate the cyclic AMP inside some of the cells along your brush border to put too much chloride into the intestine, and water follows, sodium follows because sodium loves chloride, and so you have massive diarrhea, you lose too much water, and you also lose too much salt, and you either die of dehydration or hyponatremia because you don't have enough sodium anymore. And initially, they would give them water. They knew that they were becoming dehydrated. They would give them water, and they died. And then they discovered that a lot of salt was being lost, so they gave them water with salt in it, and they gave it, and they died. And they discovered that there was a transporter that used sodium and glucose, and they added sugar. They basically gave them Gatorade, sugar, and sodium. They went through the transporter, they pulled the water by osmosis, and they started living. So these transporters that we've talked about are not just something you got to learn in school. They actually make a difference. Carbohydrates. Remember that the other thing you secrete in the stomach is intrinsic factor. It has to bind B12, so it can go bind in the ileum and be reabsorbed in the ileum. That's the only way you can absorb B12. If you can't make intrinsic factor, you have to get B12 shots, because eating it won't help. Remember the process of digesting fats, which is a little different from the other guys. You have to emulsify the fat with bile, and then you digest it, you reform it into micelles, and then it moves across, becomes chylomicrons, and moves into the lymphatic system to get back into the system. That overall process, the bile stays in the intestine, is used over and over again, and then it gets reabsorbed in the ileum also. So the two B's, bile and B12, have to be reabsorbed in the ileum. Remember the migrating motor complex, how motilin between meals will cause the migrating motor complex to move everything along better. Remember those other reflexes like the gastrocolic reflex. You put a lot of food in the stomach, it signals the colon to increase defecation so that you have room for the food to move in. So you have a variety of reflexes between one part of the GI tract and the other, sent either through that myenteric plexus of the contraction, or the submucosal plexus of secretion, or in some cases of a reflex. If it needs to go really far, it can go outside the intestinal system and then back into it at that far end, so it can have a quicker action. Know the difference between peristalsis, segmentation, haustration, mass movements, the kind of movements we get in the small intestine versus the large intestine. know what mainly the large intestine does. It mostly squeezes out water so that it receives liquid chyme and it comes out solid if everything's working right. Remember that if you inflame the intestine, it causes those villi to swell up so that nothing can get between them. You don't absorb very well and you get diarrhea. When you get diarrhea, you lose water, you lose electrolytes, you lose nutrients, and you can lose bile. You lose a lot of bicarbonate that you should be reabsorbing, and so you become acidotic. Remember, with our acid-base balance thing, all of those can happen with diarrhea. Lactose intolerance, sucrose intolerance, celiac disease, which is gluten intolerance, or you can have Crohn's disease, which is the same type of symptoms, but it's an autoimmune effect. Thermoregulation. Understand how the hypothalamus has sensors coming from the skin and from the deep tissue to tell it whether your temperature needs to be altered. And especially the ones coming from the deep tissue, because your core temperature needs to stay pretty close to 37 degrees Celsius. And if it goes up a little bit, you're going to sweat to bring it back down again, sweat and vasodilate. If you get cold, if your core temperature drops, you're going to shiver and vasoconstrict to build your temperature back up again until you reach that set point that the hypothalamus has established. Know how fever works, where if the hypothalamus is stimulated by cytokines like leukotrienes, interleukin-1, it'll know that you're fighting a bacteria or viral infection, and it'll move its set point up so that you shiver to bring your temperature up to try to kill the bacteria. At the end, it drops the set point back down, and you sweat to bring your temperature back down to normal again. Kidneys, know the parts. We've done this the most recently, before the reproductive stuff anyway. Certainly know the arcuate artery, the afferent arteriole, into the glomerulus, which is inside Bowman's capsule, and then it, goes back out the efferent arteriole. The constriction or dilation of those controls the blood flow and the pressure inside Bowman's capsule, inside the glomerular capillaries, and helps determine how much you're going to filter out. So more flow will give you more P, but also more pressure inside will give you more P by increasing the filtration fraction. The primary filtrate, which is essentially plasma without the large plasma proteins in it, goes down the proximal convoluted tubule and straight tubule, the loop of Henle descending ascending, the distal straight and convoluted tubule, and the collecting ducts. Okay, and what happens at each stage? Well, the proximal tubule, you reabsorb more than half of everything you're going to reabsorb right away, especially things like glucose and amino acids. Then you reabsorb mostly water, mostly electrolytes, and then you fine-tune it using the principal cells that respond to ADH and aldosterone and the like, to either open or close aquaporins, to speed up or slow down sodium-potassium pumps so that you pump more sodium back into the blood, reabsorb, or less, and therefore more water, or you open aquaporins so more water can be reabsorbed, or less. And then what you pee out is excreted, not secreted, but excreted. Secretion puts it in there in the first place, but it can be reabsorbed. What's excreted is what leaves your body. the high pressures in the glomerulus compared to peritubular capillaries so that you filter there and you reabsorb here. It's kind of like at each end of a capillary bed anyway, but even more so. You filter a whole lot at the glomerulus, you reabsorb a whole lot at the peritubular capillaries. Remember, ADH is required to reabsorb pure water. It opens aquaporins and allows water to move more freely back into the blood. It causes you to reabsorb pure water and therefore lower your plasma osmolarity. So ADH is a real strong osmolarity controller in your body. Lack of ADH is called diabetes insipidus. You pee too much pure water, a lot of very dilute urine, because you're mostly losing water, not other stuff that way. And then our reproductive system, we've just done that, so you can maybe watch those lectures a second time to be sure about that, and look at your practice questions. Know the steps in differentiation. Know what happens if you make testosterone or don't to retain the male stuff or not. Know what happens if you make anti-Müllerian hormone to retain the female stuff or not. Know what has to happen for that male fetus. First, it has to be a genetic male to be a functional male fetus, but then it has to get testosterone and anti-Müllerian hormone to have normal development of the male stuff, but not the female stuff. Know the hormones as they move through your life cycle, the spike of testosterone when you're a fetus, the spike at birth, stays low till puberty, then it goes up and stays up pretty high most of the rest of your life, starts dropping off in your 70s and 80s. Know the functions of the seminiferous tubules versus the epididymis versus the vas deferens, the seminal vesicle, the prostate gland, the parts of the urethra, the penis, the corpus spongiosum, corpus cavernosum, the urinary meatus or opening, the prepuce, the foreskin. Know the parts, and you will have just done those on the lab anyway, but know their function as well. Know that in the male, Leydig cells make testosterone. Know what that does. Know that a lot of it converts to dihydrotestosterone, which has a lot of effects on external genital development. Know the cycle for the female, the monthly cycle, where you start out with a ton of eggs as a embryo, you drop down a lot by birth, you drop down even more by puberty, and then every month a group of 6 to 12 of them try to make it. Only one of them does, unless two do and you have twins. But generally only one does. Usually you've got 2 ovaries. This month you get 1 from this ovary. This month you get 1 from this ovary. If you get 1 from each ovary, you're likely to have twins. Okay, they would be fraternal twins. They would be 2 different eggs, not one that splits and then instead of staying together splits all the way apart and then develops into 2 identical twins. If you get 2 separate eggs getting fertilized, then you have fraternal twins. It can be 1 male, 1 female. Okay, the increase in LH causing the hormonal changes, the theca cells are like the Leydig cells, so they make a lot of testosterone, which converts to estrogens. They form the corpus luteum around the egg, which then is left behind when you ovulate and starts producing a lot of progesterone, which is going to maintain the endometrium. The egg goes and has to get fertilized in the next 24 hours or so that it has time to split and split, become a morula, and then move in and implant in the wall of the uterus. The the LH spike that really causes the ovulation. They increase in estrogens for a while that will cause vaginal secretions and increase in activity and thickening of the endometrium, and then progesterone, which maintains the endometrium and doesn't let you menstruate. If you don't do anything from that point on, the progesterone will drop and you have your menstrual cycle and start over. If you fertilize the egg and it implants and you start making HCG right away from that growing placenta, then you maintain the progesterone, you maintain the endometrium, and the pregnancy is successful. Know how that you also make a couple of other hormones like placental lactogen, or placental, lactogen is what it's called, but it stimulates the female breast tissue to develop, so the woman's breasts develop during the pregnancy, so she'll have plenty of milk when the baby's born. Know how the parturition happens, largely because of the at the time there's so much stretch in the uterus, it will begin to contract. At least partially, the stress of contraction causes a release of cortisol, the stress hormone, which causes oxytocin to be released, which causes another contraction, which causes the stress hormone, and so forth. That positive cycle, the positive feedback mechanism that results in childbirth. Increased prolactin and estrogens at that time will produce milk in the mother's breasts, also stimulate the baby to perhaps leak a little bit of milk out of his or her breasts right at childbirth. The initial milk that comes out, colostrum, which doesn't have much fat in it, so it's not really white yet. The fat is the white part of milk. That comes a little bit later. See if there's something I really forgot. That should pretty much cover it. If you have any questions, I think I'll put a discussion board up for the between now and the final exam that lets people ask questions, answer each other's questions, see my answers, so that I don't have to answer every question for every individual 2 or 3 times, and it will just be called discussion for final. I'll go ahead and put that up. You can use it or not as you like, but if you have any questions, that'll be a good place to ask it. I'll check it at least once a day or a couple of times a day. The final exam will be put on your Canvas site on Monday and will be available till Thursday evening. So, as usual, you'll have Monday through Thursday evening to take the exam. I should have all of your grades posted by Friday evening. So, let me know if you have any questions. Take care.